Good morning, dear students. In today's class, we shall study an important structure in the GIT, that is the pancreas. We shall see what are the different secretions from the pancreas which is going to help in the digestion and absorption of the food that we consume. Okay? Before we get into the functions of pancreas, we shall see little bit about the anatomical aspect. The pancreas is a compound gland. It measures about 15 to 20 centimeters. It is J. It looks like a J shape and weighs about 90 grams. Coming to the functional anatomy, it is present in the abdomen. It is present in the abdomen towards the left side, right? You can see here it is attached to the, or it is very much near to the second part of the duodenum. It is present just next to the second part of the duodenum. Coming to the anatomical parts, it has head, body and tail. Physiological parts of this pancreas, it has exocrine part and the endocrine part. We shall look at the structure of the exocrine part of the pancreas. Here we see, can you see here? This is what you see is the glands which is going to secrete various exocrine. Exo means something which is going to be released from outside. It is going to be released into the ducts. We call it as exocrine glands. We also have endocrine glands in the pancreas. This endocrine glands contribute for various hormones. In GIT, we are concerned with the exocrine part of the pancreas. Okay, these are the glands. These glands will secrete various enzymes which is going to be poured into this duct, and this duct is going to put all the secretions into the second part of the duodenum. Can you see here? All the secretions will be poured into the duct, and this duct will open up in the second part of the duodenum. I hope you all know what is duodenum. Duodenum, it is the structure which is present, which is in continuous with the stomach, right? After stomach, we have the duodenum. In the second part of the duodenum, these pancreatic ducts will open. So all the secretions will be poured into this part. Okay. Now, this duct, we have various smaller ducts and then we have the bigger ducts. The bigger ducts are the intercalated ducts. These are the branches ones, are the intercalated ducts. They in turn form interlobular ducts. Interlobular ducts. All this will pour into a main duct. Can you see here? This is the main duct. It is like a highway. So this main duct is called as main duct of pancreas, called as duct of Wilson or Wilson duct. I repeat, this duct is called as duct of Wilson. There is one more smaller duct here. You can see this is called accessory duct or the duct of Santorini. So I repeat, if you can see here, these are the exocrine part of the pancreatic glands. See here, these are exocrine glands. These are exocrine glands. These exocrine glands are going to secrete secretions pouring into the interlobular duct and intercalated ducts, interlobular ducts. All these secretions are going to be poured into this main duct called as the duct of Wilson or Wilson duct, which is going to open up in the second part of the duodenum. All the secretions will be poured here in the second part of the duodenum. And here the function of these glands, the enzymes which are poured here, they will help in the digestion and absorption of various substances. Okay. Now, this is one more duct called as accessory duct or duct of sanguinity. These smaller glands are the endocrine glands. These endocrine glands are responsible for secretion of hormones, which we will study in the endocrine section. Okay. Here we will study mainly the exocrine glands. What are the secretions which are being poured from the exocrine glands? Okay. Pancreatic juice is a transparent, colorless fluid. It is similar, the tonicity is similar to that of plasma. It is around 1200 to 1500 ml per day. The specific gravity is around 1.01 to 1.018. And pH, this is very important. The pH of pancreatic juice is very high. It is 7.8 to 8.4. It is alkaline in nature. Why it is 7.8 to 8.4? Why it is so much alkaline? Because of abundant bicarbonate secretion. How much it is secreted? Around 113 milliequivalents per liter will be present. Whereas in plasma it is just 24 milliequivalents. In blood it is just 24 milliequivalents. Whereas in pancreatic juice it is 113 milliequivalents. And the flow rate at resting state, the pancreatic juice secretion is around 0.2 ml to 0.3 ml per minute. So in 1 minute, 0.2 to 0.3 ml will be secreted from the pancreas. This much amount of pancreatic juice will be secreted. This secretion depends upon the state, that is the feeding state or the empty state of the duodenum. Okay. Now coming to the composition. What is this pancreatic juice made up of? 99% of it is water. Only 0.5% of it is solids. 99.5% water, 0.5% solutes or the solids. Solids include both organic as well as inorganic substances. So what are the different organic and inorganic substances? The pancreatic juice contains 99.5% water and 0.5% of solids. This also includes organic substances and inorganic substances. Inorganic substances are various ions. Sodium ion, calcium ion, potassium ion, like this. All the different ions are included in the inorganic substances. Whereas organic substances include the enzymes. These are the enzymes which are going to help in the breakdown of the food products. That is, it is going to break down the protein, it is going to break down the lipids, it is also going to break down the carbohydrate. So, what are the different proteolytic enzymes? They are trypsin, chymotrypsin, carboxypeptidase, nuclease, elastase and collagenase. I repeat, you can note it down. The different proteolytic enzymes present in the pancreatic juice are trypsin, chymotrypsin, carboxypeptidases, nucleases, elastases and collagenase. The different lipolytic enzymes means these are the enzymes which is going to break down the lipid molecules. The lipolytic enzymes are pancreatic lipase, cholesterol ester hydrolase, phospholipase A, phospholipase B, colipase and bile salt activated lipases. These are the different lipolytic enzymes. Remember, pancreatic juice has abundant amount of proteolytic and lipolytic enzymes. Various types of enzymes are present. Whereas only one amylolytic enzyme, that means only one enzyme is going to break down the carbohydrate. If you remember, there are three major compounds present in the food. That is, one is the carbohydrate, second is the protein, third is the lipids. Right? So pancreatic juice has enzymes which is going to break down all three of them. So this is a major secretion which helps in digestion process. Okay, we'll see each of these enzymes in detail, how it is going to function. Yes, pancreatic juice is very rich in carbohydrate, right? So this high bicarbonate content. This high bicarbonate content makes the pancreatic juice highly alkaline. What is this? What is the advantage of this highly alkaline content of pancreatic juice? One is it protects the intestinal mucosa. The food present in the stomach, what we call it as the bolus, which enters into the stomach. In the stomach, it is formed chyme. Right? This chyme mixed with acid is highly acidic in nature. So once this acid chyme enters into the duodenum, this can actually damage the mucosa of the duodenum. So in order to protect that, high bicarbonate content of pancreatic juice will neutralize this acid chyme. So it acts as a protective mechanism to the intestinal mucosa. So high bicarbonate content makes the pancreatic juice highly alkaline so that it protects the intestinal mucosa from acid chyme by neutralizing it. One more important function of bicarbonate-rich secretion is it provides the necessary pH for the activation of various pancreatic enzymes. Okay, so this pH is required for activation of these enzymes which are released from the pancreas. Now let us see what are the different pancreatic enzymes. What is their function? Pancreatic alpha amylase. Remember the word amylase. Amylase means the enzyme that is going to break down the carbohydrate. Okay. So this pancreatic alpha amylase is an active form. It is secreted itself in the active form. This is the only amylatic enzyme present in the pancreatic juice. So the pancreatic juice contains only one type of enzyme which can break down the carbohydrate. Just like salivary amylase which is secreted in the mouth, pancreatic amylase also converts starch into dextrin and maltose. That is, bigger molecule of carbohydrate is broken down into two molecules. Into the dextrin and maltose. Now, this is about the pancreatic alpha amylase. Then we have pancreatic lipases. Lipase means those enzymes which can break down the lipid substances, fat substances. We call them as lipases. So, what are the different enzymes which can break down the fat substances secreted from pancreas? They are, one is pancreatic lipase, second is cholesterol ester hydrolases. Cholesterol ester hydrolases. Then we have phospholipase A2, which is secreted in the inactive form, but it gets activated in case of acute pancreatitis. This is a disease condition. These are the lipases for the lipid dissolving enzymes secreted from the pancreas. I repeat, it is the pancreatic lipase and cholesterol ester hydrolases. Then, coming to digestion of proteins. So, we finished with digestion of carbohydrate, which is the enzyme which is going to digest the carbohydrate molecule secreted from pancreas. It is pancreatic alpha amylase. Which are the enzymes which is going to digest lipid molecules secreted from pancreas? Pancreatic lipase, cholesterol ester hydrolases, and phospholipase A2. Now we'll see how protein is going to be digested by the enzymes of pancreas. The major proteolytic enzymes of pancreatic juice are trypsin and chymotrypsin. These are the two important ones. Other proteolytic enzymes, proteolytic means it is going to lyse the proteins. Protein moiety. Protein is, what is protein? It is a combination of various amino acids, chain of amino acids, about hundreds or fifties or thousands of amino acid molecules joined together to form protein. So these protein moieties, that is the amino acids, are broken down by these enzymes. What are these? They are trypsin and chymotrypsin. Other proteolytic enzymes are carboxypeptidases, nucleases, elastases and coagulases. Right? Let us see what are these pancreatic proteases. Those pancreatic enzymes which are going to cleave the amino acid chains in between, we call them as endopeptidases. We call them as endopeptidases. They are endo means inside. They are going to break down the inside bonds. We call them as endopeptidases. They are trypsin, chymotrypsin and elastase. Whereas exopeptidases means it is going to keep the last amino acid molecule. It is going to come from periphery to center. Last amino acid molecules are going to be cleaved or lysed by exopeptidases. These exopeptidases are carboxypeptidases A and E. And we also have nucleases and coagulases. Those which are going to cleave the nucleic acid, we call them as nucleases. Those which is going to cleave the collagen moiety, we call them as coagulases. All these enzymes, one important thing what you have to remember in pancreatic proteases are, all these enzymes are secreted from the pancreas, poured into the duodenum in the inactive form. This inactive form of all these enzymes, once it enters into the duodenum, in the duodenum they will get activated. So how it gets activated? This is because of the enzyme present in the intestine called endopeptidase. I repeat, an enzyme present in the intestinal peptide, intestinal enzyme, intestinal proteolytic enzyme is going to first activate only one, that is trypsin. This trypsin, once it gets activated, that will convert all other inactive enzymes into active forms. Trypsin will convert all other inactive enzymes into active forms. Okay? So why this is important? Because this trypsin, if it gets activated inside the pancreas itself, it will cause something called as auto-digestion of the pancreas. Because even pancreas are made of proteins, isn't it? All the cells have proteins inside them. All the enzymes have protein inside them. So this trypsin will cause auto-digestion of the pancreas. So in order to prevent this auto-digestion, we have something called as trypsin inhibitor inside the pancreas. So inside the pancreas, this trypsin will not get activated. It will get activated only when it enters into the intestine. Now let us see what are the functions of these proteolytic enzymes. One is digestion of proteins. Trypsin is the most powerful proteolytic enzyme. It is going to cause digestion of proteins. It is an endopeptidase, that is, it breaks the interior bonds of the protein, inside bonds of the protein molecules. It converts proteins into proteoses and polypeptides. By cleaving the inside bonds, it will convert one big chain into smaller chains. Smaller chain of amino acid call them as proteoses and polypeptides. This is one function. Second function is they cause curdling of milk. It converts caseinogen in milk to casein. So how curd is formed from milk? Because of the action of these enzymes. Then blood clotting. It accelerates the blood clotting mechanism. It also activates other enzymes of pancreatic juice. The trypsin also activates other enzymes of pancreatic juice, that is chymotrypsinogen to chymotrypsin, pro-carboxypeptidases A and B into carboxypeptidases, pro-elastase is converted into elastase, pro-polypase is converted into polypase. So all these are inactive enzymes. Can you see? These are inactive enzymes. The other form, that is chymotrypsinogen, is converted into active form, that is chymotrypsin. Like this. So trypsin is going to convert all other inactive enzymes secreted from pancreas into active enzymes. Trypsin also activates collagenase, phospholipase A, phospholipase B. It also has autocatalytic action. Once it is formed, trypsin will convert more and more trypsinogen into trypsin. All these functions are performed by the proteolytic enzyme trypsin, which is secreted from the pancreatic juice. Coming to the mechanism of secretion of pancreatic juice. Functions of pancreatic juice, it is going to help in the digestion process because it has alpha-amyloidic enzyme. Alpha-amyloidic enzyme is going to digest carbohydrates. It's going to break down the bonds, convert starch into dextrin and maltose. It is going to convert lipids substances into smaller fatty acids. It also converts protein molecules into peptones and polypeptides. Also, exopeptidases can convert polypeptides into amino acids. So, complete digestion of protein can occur by the help of pancreatic enzymes of pancreatic juice. It also has a neutralizing action because it is rich in bicarbonate. This bicarbonate will neutralize the acidic kind. Now, what is the mechanism of pancreatic secretion? You have the acinar cells. Acinar cell is nothing but the cell of the gland. Right? So, these cells are going to secrete the pancreatic juice rich in enzymes. Now, how bicarbonate is secreted? You have the blood vessels, this is the ductal cells, and this is the lumen. Lumen means the hollow structure. GID, hollow structure of GID. Water and carbon dioxide enters into the ductal cells. Carbon dioxide combines with water with the help of carbonic anhydrase. There is an enzyme called carbonic anhydrase, which is going to combine carbon dioxide with water to form bicarbonate. Bicarbonate, H2CO3, is now going to dissociate to form bicarbonate and H plus ions. Carbonic acid is now converted to bicarbonate and H plus ions. This bicarbonate is going to be secreted into the secreted into the lumen. From the ductal cells, it will be secreted into the lumen. So, this is how bicarbonate is secreted into the pancreatic juice. Coming to the regulation of pancreatic secretion. The basic stimuli for pancreatic secretion is the enzyme or the hormone which is going to stimulate pancreatic secretions are acetylcholine, cholecystokinin and secretin. Acetylcholine, cholecystokinin and secretin. These are the hormones. The cholecystokinin and secretin are the hormones which is going to increase the pancreatic secretion. So there are three different phases of pancreatic secretion. One is the cephalic phase, second is the gastric phase, and third is the intestinal phase. Cephalic phase means the very thought of food increases the pancreatic secretion. Or sight of food, you see the food, you smell the food, or you just think about the food, there will be increase in pancreatic juice secretion. That is called as cephalic phase. That is cephalic phase of pancreatic secretion. Then we have the gastric phase. When the contents of food enters into the stomach, what we call it as chyme. Okay, when the contents of food enters into the stomach, here in the stomach we have two important hormones released. One is the acid from stomach releases secretin from the walls of the duodenum. Secretin is secreted by acid of the stomach, enhances the secretion of secretin from the duodenum. So one is secretin. Then the fats and amino acids also cause release of cholecystokinin. So these are the two important hormones which is going to enhance pancreatic secretion. Secretin and cholecystokinin. I repeat, secretin, cholecystokinin. These two are going to increase pancreatic secretion. They have two different roles. Okay. The secretin is going to increase the secretion of pancreatic fluid and also the bicarbonate. Whereas cholecystokinin is going to cause secretion of the enzymes. I repeat, secretin is going to increase pancreatic fluid and bicarbonate secretion. Cholecystokinin is going to enhance the secretion of various enzymes required for digestion. Okay. Now, this and also various stimulation increases the release of enzymes in the acidity. So these are the stimulators of secretion from the stomach. This is what gastric phase of pancreatic juice secretion. So when food enters into the stomach, when it forms acid chyme, the acid chyme is going to stimulate the secretion of secretin and cholecystokinin, which is in turn going to increase the pancreatic secretions. So this is all about how the pancreatic enzymes. Till now we have studied how the pancreatic enzymes are going to help in the digestion process. We have also learned about how the pancreatic juice secretion is going to be enhanced, right? So we learned about the cephalic phase and gastric phase. Intestinal phase, as the acidic chyme enters into the duodenum, the secretion of secretin enzyme, again secretin hormone again is going to be increased, which is going to enhance the pancreatic juice secretion. We call it as intestinal phase. So three phases of the pancreatic juice secretion: cephalic phase, thought of the food, smell of the food, the vision of the food; gastric phase, when food enters into the stomach, release of acidic chyme, increases secretin and cholecystokinin; intestinal phase, when the acidic chyme enters the intestine, again there is increase in pancreatic secretion. So these are the regulation of pancreatic juice secretion. Now we will see some of the applied aspects of pancreatic insufficiencies. The causes for pancreatic insufficiencies are acute pancreatitis. This is acute inflammation of the pancreas, chronic pancreatitis, cystic fibrosis, pancreatectomy, that is removal of the pancreas, alcohol-related pancreatic disease. We will see some of them. These are some of the important diseases which is going to affect the pancreas. Acute pancreatitis, it is an acute inflammation of the pancreas. So whenever there is acute inflammation, there is release of trypsin. Trypsin gets auto-activated and there can occur auto-digestion of the trypsin. autodigestion of the pancreas by the trypsin. Chronic pancreatitis usually caused by obstruction of the pancreatic duct. Cystic fibrosis is a hereditary, it is a inherent, it is a disorder which occurs by birth. Any disease which is going to affect the pancreas chronically, we need to remove the pancreas and called as pancreatectomy. Increase in alcohol consumption again can lead to acute and chronic pancreatitis. Coming to pancreatic function test, how do we assess the functions of the pancreas? One is by collection of pancreatic juice and measuring how much quantity of pancreatic juice is being secreted. And then analysis of pancreatic juice that has been collected after direct stimulation of pancreas. We do two tests called as secretin test, combined secretin and cholecystokin. What we do is we inject secretin hormone and see how much of pancreatic juice secretion will be increased. One more is we inject secretin and cholecystokin and see how much of pancreatic juice is going to be secreted. Then analysis of pancreatic juice which is already collected after indirect stimulation of pancreas. This test is called as Lung test. We can also do the analysis of products of digestion, that is fecal fat excretion test, fecal nitrogen excretion test, tripeptide hydrolysis test, dual label Schilling test. All these are different tests to analyze the products of digestion. Adequate amount of pancreatic juice will cause proper digestion of fat and proteins. If there is improper digestion of fat and proteins, if there is excretion of fat, more and more excretion of fat, more and more fecal nitrogen excretion, then pancreatic insufficiency is said to be existing. Then we can estimate serum amylase. This is the most common test done to detect pancreatic insufficiency. Estimation of serum amylase. Serum amylase normally is 50 to 120 units per liter. This is all about today's class. Until now we have studied that what is pancreas, what are the different types of pancreatic secretions, exocrine pancreatic secretions and endocrine pancreatic secretions. Then we studied what are the different exocrine pancreatic secretions which is going to help in the digestion process. Then we studied what are the regulators of pancreatic juice secretion. Then we also studied some of the applied aspects and pancreatic function tests. So this is all for today's class. Any doubts you can put up in the group.